All right, so hi everyone. Welcome to the ECCB tutorial. This is the session for full-length RNA-Seq using PacBio long reads. And I will share my screen again. No, actually I won't first because I want to introduce the instructors first. And also during this time, at least for the first minute or two, feel free to turn on your webcams because, you know, if this had been a physical conference, we would have been able to see you. If your internet speed will allow, we would love to see your faces, at least for the next few minutes.
No, they can't turn on the camera.
Oh, they can't turn on the camera?
No, just the mic. They can turn on the mic, but not the camera, no.
Oh, yeah. Yeah, so I can see some people turn on their cameras.
Oh, yeah, they can.
No, they can.
Yeah.
Awesome.
Cool.
Yes.
All right, so while you turn on your webcams, I will do some introduction. So I'm one of the instructors. My name is Liz, and I'm a principal scientist at PacBio. And the instructors we have today will have each of them maybe just give a brief intro of themselves. Is Ana, Angeles, Fran, and Kay. So I'll start with myself. I'm a principal scientist at PacBio. I'm one of the people who kind of created the algorithm for ISCB, which is what is one of the software that you're going to use today. It comes with PacBio's official software suite. And then I'll let Kay introduce herself.
Thanks, Liz. Hi, my name's Kay. I'm a bioinformatics field application scientist based out of the DC area. So I'm from PacBio, and I'll be here to help you guys answer questions if you are going through the tutorial with Liz and have questions as you're going along. So it's nice to meet you all.
Okay, thank you. My name is Ana Conesa. I'm a professor at the University of Florida, and I had developed several tools for the analysis of long reads, mainly tools that go downstream to ISO-Seq3. So once you learn to pre-process your transcript with that software, then you can go into the actual quality analysis and interpretation of the results with the tools that we have developed in my lab. So I will be presenting that and then helping you guys when you have questions to answer them.
So should I go now? My name is Angeles. I'm a PhD candidate at Ana Conesa's lab, and I'm based in Valencia. And while I do my PhD thesis on single-cell RNA-Seq, I've been doing a lot of work on tapas recently and participated in validating the application a year or so ago. So I'm going to be teaching that part of the tutorial and the hands-on, and here to answer any questions you have regarding the application.
Fran?
Yes.
Oh.
Okay,
Angeles needs to mute, not the other way.
So
We have to record it.
I'm going to remove sound and mic, and you turn on your sound and mic.
So hello, I'm Fran, and as Angeles said, we are in Valencia. I'm part also of Ana Conesa's lab, and I'm one of the developers with Liz and some other people of Scanty and part of IsoAnode, and I will explain that part of the functional isotranscriptomic pipeline in this tutorial. So if you have questions now during the tutorial or afterwards, I will be happy to answer all of them.
Yeah, so a little bit of a housekeeping is, as I know some of you were joined on ISO-Seq Slack, thank you very much. However, during this actual session, we will not be using Slack. And I guess you are all muted, so you cannot talk to us either. So please use the ECCB platform. So if you go to your ECCB platform, first of all, there is a poll. We've asked you, what is your expertise? Have you completed the ISO-Seq installation? And if you have questions, feel free to use the live Q&A. And if you look at the discussion forum, I have already posted an updated Dropbox link. If you go to that link right now, so you know what, I'm going to show because it is important that you're able to do this while, maybe while I do the intro. So you guys can see my screen now, right? Okay, so if I go to this Dropbox, and I need to make sure I don't click on anything else. Okay, so if you go to this Dropbox right now, you'll see that there are 2 text files. They're very small. If you go to click and download this, you'll be able to see this is the actual instructions that we are going to use for the first hands-on. If you have already, you should have already installed ISO-Seq 3 in a Conda environment, either on AWS or your local server. So, if you did AWS, this is similar to what you would be doing. If you are on a local machine, you should be SSHing into it right now and activate your Conda environment. And the more important thing is we do need to download the test data for this workshop. I know it says ISMB, but that was because we are reusing the data. You should probably log in and do the wget for these 4 right now, mainly because I did not partition the reference genome to a smaller toy set. So it takes maybe a minute or 2, depending on your internet speed, to download HG38. So if you have not done so, if you can, while I'm doing the intro or while I'm doing kind of the explainers, please log into your Conda environment and download these 4 files. Okay. Okay, so with that, I'm going to go through the agenda. So right now, and where's my phone? I can't find my phone, so it's okay because I can't see the time. But this is the session. So Anna and I will give an introduction, and then I will walk you through the hands-on section. And I'm Liz, and Kay will also help you. Again, use the live Q&A during any time of the discussion if you have questions. Kay, who is the PacBio FAS, will help you answer them. We have a coffee break for 10 minutes. And then Fran and Angeles will lead the Scanti tutorial. Then we have another 10-minute coffee break. And then Angeles and Fran will talk about tapas. Okay, so I will start with my introduction. So I'm giving you the intro to what the ISO-Seq method is. So what is ISO-Seq? Really, ISO-Seq is something that is a little different from traditional RNA-Seq that maybe you're used to, is that in traditional RNA-Seq, the transcripts are fragmented into smaller pieces and then sequenced with short paired end reads. What this means is no read covers the entire transcript in full, and then hence identifying the full-length transcript is a computational assembly problem. But as many of you know now, this computational assembly is very challenging, and sometimes even with the best software, you simply cannot get the right answers because of the splicing complexity. In contrast, the ISO-Seq method on the PacBio systems eliminates the assembly problem by simply sequence the full-length cDNA. And then you sequence the entire read, no assembly is required. This is possible because the PacBio read lengths are much longer. It is also accurate because we are sequencing the same insert multiple times. So actually the actual sequencing technology is done in a circular fashion. So we have this double-stranded cDNA. This is where your transcript is. And then when we sequence it, we're going to go around it many, many, many times. So if you're familiar with consensus, the concept of consensus calling, we're actually giving you for every read a very, very accurate read that is reread of the same insert many, many times. And for the purpose of doing RNA analysis, this accuracy is high enough that you would be able to call SNPs, call indels, look at alternative splicing, or if you're doing single cell, you'll be able to call UMIs and barcodes. And we're not going to talk about the library preparation for this workshop, but just to say that the ISO-Seq workflow is end-to-end. The set library prep is 1 day. Sequencing is also done on 1 day. And then we're going to talk about the analysis today. So I'll give you a quick kind of survey of what people have been using ISO-Seq for. The first one in a plant and animal space. This is a full-length isoform phasing study with maize. The setup is that we had 2 inbred pure parental lines of maize, and then they're cross hybrids, where we swap what who the male and the females are. And then the goal of this study was to identify allele-specific isoform expression using PacBio. The concept, we're not going to talk about isophase today, was that if you take it through the ISO-Seq analysis, you have all the information already for you to do phasing. So on the left side is basically what we're trying to learn today, at least for the first part, is you start with the PacBio data. This is where we're going to teach the ISO-Seq analysis. We align it to the genome, filter some artifacts. In fact, we do use Scanty at this step. And then we get these high quality, non-redundant full-length transcripts. Then we can go back and look at the individual molecule information and then look for isoform information at the allele level. So just to show you an IGV screenshot, so this is using the IGV browser. I don't know what you guys are familiar with for visualizing splicing, but either IGV or UCSC browser are very popular tools. Using the IGV browser, what we're showing here is the 4 different samples, so the 2 parents and their S1 hybrids. And we see in this first example is that we're aligning to the B73 reference genome. So when you look at the B73 ISO-Seq data, you shouldn't really see any SNPs because this is against the same reference. But when we do K11, we see where all the SNPs are. When we then go look at the F1 hybrids, what's interesting is that for this one, where the maternal allele is Ki11, only the Ki11 allele is expressed. We do not see the expression of B73, even though it is the paternal allele. However, when we switch who the maternal allele is, we now see that in this F1 hybrid, only the B73, which is the maternal allele for this hybrid, is expressed. In another one, we're able to see not only parent-specific expression, but we're also able to see parent-specific isoform expression. So in this case, the difference between B73 and Ki11 is that the last exon is spliced out in Ki11. When we go look at the 2 F1 hybrids, what we see is that actually this time, both alleles are expressed. However, it inherited the parent-specific isoform. So for the allele that comes from Ki11, we see this is still spliced out. And then moving into the human space, ISO-Seq was also used to look at double mutations in the PIK3CA gene. The background for this is the PIK3CA mutations are present in ER-positive, HER2-negative cancers. During the drug trial for Alpelisib, the researchers discovered that a subset of the patients were significantly benefiting from the drug, and that they had suspected that this might have something to do with double mutations. However, due to the sequencing technology limitations they had at that time, they weren't able to discern whether the mutations were trans, meaning that they were 2 mutations on 2 separate alleles, or cis, meaning that they were 2 single mutations on the same allele. And this is the reason they weren't able to do this with either Sanger sequencing or short-read sequencing was because the distance of the mutations were actually quite large. So here are the common double mutations, and you can see their cDNA distance were somewhere between 270 bases to 1.5 kilobases. That means a lot of them were not coverable by even Sanger sequencing. However, using PacBio, all of them would be able to be phased. What the researchers then did was they sequenced 6 of the cancer patients, and then they showed that indeed in all 6 patients, the double mutations were in cis. Well, they then went back to look at a previous clinical trial, and they were separating out the benefits of the mutations. They showed that the multiple mutation population had a significant benefit over placebo than the single mutation drug population. So this this study had far-reaching consequences because it is not a small portion of cancers that have double mutations. In fact, it is estimated that up to 20% of all PIK3CA cancers have double mutations. And if it is true that double mutations in cis have better drug response, it really does have clinical actionable items moving forward. So if you're interested, I have interviewed the author on this study, and you can go read it at my Medium blog. And I'm going to go very quickly through the software because we're going to talk about it in the hands-on session. But this is basically what the computational tool environment looks like. So we have the sequencing, and then we're going to talk about the ISO-Seq analysis. And Fran and Angeles will walk you through classification, annotation, and differential analysis. And really, the tools that are going to suit you, it really depends on what your purpose is. If you're doing genome annotation, some of our plant and animal customers would be using things like Maker and Augustus. If you're doing allele phasing, you might be using isophase. And if you're doing differential expression, you would be following the pipeline that we are doing today. And if you want to learn more about the ISO-Seq method, there's a lot of ways to reach us. In the Dropbox that I have shared with you at the beginning of the session, all the slides and materials from today will remain in the Dropbox after the tutorial. With that, I think it is Anna's turn to give her intro. Anna, you have to unmute.
Oof, I have too many windows here.
Yeah.
Okay. Can you see now my full screen? Yes, I guess.
Yes, I
okay.
can.
Okay, great. So hello everyone again. So Liz introduced like applications of the long reads and a kind of overview of what we are going to be doing today. So the types of tools and analysis that we are going to be presenting relates to the downstream analysis of the ISO-Seq data. And basically what we will be introducing here is what we call the functional isotranscriptomics analysis framework, in which we are trying to address a number of questions like which are the true transcripts that are present in my transcriptome, how transcript expression changes between conditions, and what are the functional consequences of these changes. And for that, my lab created a set of tools, Scanty, IsoAnnote, and TAPAS, that are able to address these questions. So I will go very quickly through each of these three tools, and then we will be doing a tutorial on them. So what is Scanty? This is a tool for the quality control of transcriptome long reads data. So what Scanty is doing is to identify which transcripts present in the reference are also present in my long reads data. It will also help you to identify novel transcripts, and this is something that you will find in every single experiment. Novel transcripts are being expressed. It will help you to evaluate the quality of the data and remove all these artifacts that are problems of the technology or your experiment, and retain only bona fide transcripts. So basically you start with the long read transcriptome that comes from the ISO-Seq analysis. That's what is Scanty. You can somehow select and identify those that are the true transcripts, and this gives you your curated full-length transcriptome. Then we go into IsoAnnote, that is a tool for the functional annotation of these transcript sequences. So what we do here is to assign functional labels to transcript models, and these labels are both for coding as non-coding regions, like for coding could be, for example, PFAM domains and non-coding microRNA binding sites, and we can also identify, for example, nonsense mediated decay transcripts. So from the previous step, we have our curated full-length transcriptome, and with IsoAnnote, we are going to be identifying which is the coding region, which are the different domains and motifs and signals that are present in these transcripts. And finally, if you are comparing different experimental conditions and then you want to assess isoform changes, you can use TAPAS. TAPAS will help you to analyze the diversity of isoform annotations, calculate differential isoform usage, and because we have all this isoanode information, we can find the pathways and functional domains that are affected by splicing. And we can also use TAPAS to evaluate differences at UTRs. So again, we have our fully annotated full-length transcriptome, and with TAPAS, what we can do is evaluate different conditions, and for example, see that we have here a gene in which we have a change in the expression of isoforms that is different between conditions, and because we have this functional information, we can, for example, identify that this functional domain is upregulated with these functional motifs is downregulated when comparing the two conditions. So the basic overall scheme or workflow is as follows. So you start with your long reads. You use ISO-Seq 3 to create a transcriptome in the form of a GTF file. Then you can use Scanty to filter this and to create the transcriptome so that you will have a fully annotated, a curated transcriptome, which can also be annotated with isoanode. For quantification of this transcriptome, we are normally using short reads. This is what we will show here, but I think that in the future, the long reads alone will be sufficient. But then using these quantification options, you can create an expression matrix of your gene expression in different conditions. So the gene expression together with your experimental design and the annotation of the transcriptome that you obtain with isoanode, they all go into TAPAS. Sometimes for some species, we already have this annotation implemented, so you can just use it directly from TAPAS. You don't have to do this step. But then in TAPAS, we have a set of modules for the analysis that evaluate functional diversity, differential expression analysis, and splicing analysis, and the functional consequences of this. These are the papers describing Scanty and TAPAS, and this is the GitHub, so you can access these tools there. And that's my little intro. Thank you, and I hope that you will enjoy this functional isotranscriptomics tutorial. And then I think that we go back to Liz and Kay.
Yep. So my turn to share my screen again. Okay. Ooh. Okay. So let me actually go to, hold on, where did the slides go? This one. Okay, so I am being mindful of the time. I want to finish my hands-on in about 15 minutes, and you know, a lot of these tutorials, so these slides will become available almost instantly, you know, right after during the coffee break, I will put them online. So what I think I'm going to do is this deck will be available, but I'm actually going to just walk you through maybe 5 slides of it, because the rest is actually the actual commands that we're going to run anyway. So I'll just show them to you. So we are going to walk you through the ISO-Seq tutorial. Everything is documented at this GitHub link. So, you know, at any time, go visit it. And, you know, also be mindful that, you know, software do change, and, you know, for ISO-Seq, we're always constantly improving it. So even if you do have the materials from this tutorial, I do recommend always checking the PacBio GitHub link for the latest version. But I want to walk you through the concept of what we are going to do. So we have these highly accurate reads in PacBio called hi-fi reads. And what they will look like is that you'll have the primers, the cDNA primers, and the poly-A tail. And as you know, what the bioinformatics step do is we don't need the primers. You know, they were useful for the sequencing, but they're not actually part of the biology. So the first part we do is we identify and remove the cDNA primers, and we call these full-length reads. And then in the next step, we are going to remove the poly-A tails, but also sometimes there are some artifacts, you know, such as concatenators that are more than multiple cDNAs that accidentally gotten concatenated together and went through sequencing. We're going to identify them and remove these. So now we have what you should consider after the ISO-Seq refine step. clean data where every read is full length, it is oriented from the 5' to the 3' end, and it is free of concatenars. The next step we're going to do is many of these reads will actually come from the same isoform. So the cluster step, remember we don't do assembly because we have full length information. We're going to cluster them by isoform. And then we can call a consensus so that each isoform cluster only has one represented sequence. Then, even though this is optional, is if you do have a reference genome, notice that in all the previous steps, we actually do not need a reference genome. But if you do have a reference genome, we are now going to map them back to the genome, and then we can get the splicing structure. And the benefits of this software is that I detect full-length transcripts and we don't actually need the reference genome. So I'm going to skip this. And again, if you go to the Dropbox that is in your discussion session, you'll be able to download this step 1 hands-on text file. If you were not able to do the pre-installation, which I looked at the poll results, some of you were not able to, you can always go back. The Dropbox contains the instructions for installing either from AWS or on your local server. But for now, you can just follow along. So if you, I've already logged into my local account. I am not using AWS today. I'm using my local work account. And I've already activated the Conda environment. You know, the first thing I can do is I can check my software version. Ooh, sorry, clearly I forgot, I logged out. So now I'm going to copy paste these 3. And you can see that I am on ISCB version 3.3, Lima 1.11, and PBMM2 1.2. And I've already downloaded the test data. Okay. So that's this step. If you haven't downloaded the data, you can do this. And remember, the first step we're going to do is we are going to identify the cDNA primers. So if we go look at the primer file, that's what it looks like right now. So that tells me this is the 5' primer, and this is the 3' primer. So I'm going to copy paste this line and run the command. And then when it's done now, you should be able to see the first 3 lines. It looks like this, so we'll do that. And what it says is you have given me this many input of hi-fi reads. And then of that, 90% of them, I have been able to identify the 5' and the 3' cDNA primer. Okay. So after that, now we have the full-length reads. The next step, refine, what it does is I detect the poly A tails and also remove artificial concatenars. So we're going to copy paste this line. And when that is done, we will get a JSON file that tells us, okay, you have this many full-length reads, which is exactly the number from here. And I was able to identify this many that were not concatenars, and of them, this many had poly A tails. So if we do the JSON file, that's exactly what we see here. Now we're going to run clustering. So I think this step takes a little bit longer. And you can see that it is currently doing the clustering. And then when it is done, you can look at the report. And what the report says is there's, let me make this just a little bit bigger. What the report says is these are the IDs for the full-length reads, and then they belong to this cluster. So the cluster has a prefix that says transcript /0. So that means the first cluster has 2 reads and the second cluster has 2 reads. Okay, it's still ongoing. It should be almost done. And then I will also just go ahead and say what the next step we're going to do is that once we are done, we are going to align it to the reference genome, HG38. Okay, so it's done here. So you can see that there are some output files. And in fact, this is the one that you're probably going to always analyze. It's the high quality FASTA files. And you can see indeed there's transcript /0. It contains 2 reads. It's this long, and that's one sequence. So now we're going to align it to the genome. This might take a little longer because it has to, the most of the time is actually spent by Minimap2. So this is a wrapper for Minimap2. It's actually just spending time reading in the reference genome. So that's going to take a while. But once that it's done, the last step we're going to do is, well, now that we have an alignment, we are going to convert that alignment into a GFF file. And then if you look at the GFF file, you should see something that looks like this. And what that says is that the first transcript, so it always starts with a prefix PB, it's going to say the first PB, PB.1.1 is a transcript that contains this many exons, and this is the exon structure of each of them. And in fact, you can take this GFF file and load it into the IGV browser, and you'll be able to see all the alternative splicing. Okay, so I have been making really good progress. I have 5 minutes left. Maybe I'll let you work through these right now. And if you have questions, please use the live Q&A or discussion. I don't know if you can unmute, but if you can unmute and you'd like to talk, feel free to do so. Okay, I can see it's still reading the reference genome. Okay, so I'll stop sharing my screen. And let me take a look at my schedule again. So right now, okay, so yeah. So right now it is 5:35 p.m. where you, for those of you in Spain. So from now till 5:50, so 5:50 is the end of the coffee break. If you have time, you can spend time right now to continue moving with the hands-on. And you can ask us questions through live Q&A or discussion. Otherwise, I will see you back in 15 minutes, where we will do the hands-on for Sconti 3. Okay, so I see a question. Ooh, did that question just go away? Oh, okay.
Yeah, Liz clicked on the answered questions because I
Oh, I
also had
see.
the same.
Because when you answer, it goes away. Oh, yeah. So I saw one question is, how many reads are moved by each analysis step, and what is the share of remaining reads before clustering? That's a great question. It really also depends a little bit on the success of the library preparation and the sequencing. But in general, we see that after the kind of the step before clustering, you should have somewhere between 60 to 80% of the data remaining. If there's something, the library had a unusually high number of artifacts, or, the A palliative was missing, then yes, you would see less. But in general, anything above 60% is great. Anything lower, usually we would go do a deep dive and take a look at what might have happened. Okay, so I'm also taking a look at some of the other questions. Right, so how are the reads assigned when some reads can be in several isoforms, if the splicing is before or after these reads? So I did see an answer to that. I think, so Maria, you can ask me, feel free to clarify, but I think what you understand, these reads are full length, right? So first of all, they should represent the full length isoform, but if you're talking about these reads might be pre-spliced, they will, in the de novo session, in the clustering parts de novo, we don't really try to distinguish, if this is a premature mRNA or not. If they look different from the rest, they will just be clustered differently from the rest. And
I think possibly an answer here is that when then you go ahead and characterize these reads, maybe you will see that this transcript is a retaining transcript, and then you will classify as such. Yeah, and that's, for example, something that we do in Scanty. So when you have this case that the read was obtained before the actual splicing takes place, then we say, okay, this is a true molecule, right? But it's an interpretation.
Right. Yeah, I think that is basically kind of like the part. I think what I philosophically now that I have 10 minutes coffee break, is philosophically I've always thought that the point of ISO-Seq is to be as agnostic to any definition of annotation and structure, because that's what Scanty and all the rest is supposed to do, right? I think the beauty of ISO-Seq is it's supposed to work with anything. It doesn't matter if it's a plant, it's an animal or bacteria. But then you said, well, is that an intron? Well, that only matters in the context of understanding what's an exon, and only understanding the context of an annotated genome. So I kind of separate that out. But I think that's a great question, and that's what Scanty, and ISO are not going to tell you. Oh yeah, so also, but got the error message. Ooh. So when I see that you installed everything properly, but got an error message, I have never seen that. And this, when are you on AWS or on a local server? Local server. I might not be able to help you, because this, my guess is you may have to talk to your IT support about this. Okay. All right, so another question is the technology biased for transcript length, favoring short transcripts, for example? So not so much anymore. I mean, if you have been using PacBio in its previous generations, there was more pronounced what we call loading bias, but in the current PacBio system called SQL2, we are able to see transcripts lengths up to 14 or 15 KB. We were able to sequence it quite easily. And I would say that at this point, the limitation of being able to sequence long transcripts is more in the success of being able to reverse transcribe a very long transcript. So, generally we don't see anything over 20 KB. not because of the sequencing issue, but because it's very difficult to reverse transcribe a 20 kb RNA into cDNA. And as kind of, if you think about it for genome assembly, so PacBio is also used for genome assembly. In general, the libraries that we make in genome sequencing is the minimum is usually 20 to 25 kilobases, and we do not have issues with sequencing those. Yazini is asking, does the program deal with all type of alternative splicing patterns? So yes, so very good question. So in the ISO-Seq GitHub, we specify our definition of two different isoforms. It is set in stone right now, is that 2 reads are considered different isoforms if they have more than 100 base difference in the 5' end and 30 base difference in the 3' end. And internally, they cannot have more than, they cannot have a gap that has more than 10 bases. So in some sense, if you have a microexon that is less than 10 bases, it is possible for ISO-Seq3 to miscluster them together. But in eukaryotes, most exons are well over 10 bases. They're usually, I think, 60 to 100 bases. So we should be able to cover the majority. 60 to 100 bases. So we should be able to cover. So we should be able to cover. So also, if you haven't looked at the discussion, I have put all of the slides that I have just presented in the Dropbox, and this Dropbox will remain after this session.
So I am seeing the results of the poll that says that basically only half of the participants were able to pre-install all the of the software. So in this tutorial, we are not going to be showing how to install. So you can run the exercises because you already have everything installed. So if you haven't been able to install some part of the software, because all the materials and the presentations and the recording will be available, you can maybe go back later once you have the installation resolved to do your own practice on the exercises. And if you have any problems with installation, you can always use the Slack channel to ask anything related to that, so that we can help you to get everything installed.
Yes, and also we have 2 more hands-on coming, and they are not, all 3 of them are independent. If you had failed the first one, you will still be able to continue on the second one. Also, the materials will stay, and after today, if you want to keep in touch with us, there are ways. So the most convenient way to reach us would be either through the ISO-Seq Google group, which is on the last slide of my presentation, or you can join the Smart ISO-Seq Slack. I'm trying to remember, Anna, last time. Oh, I think what I did last time was I sent an invitation link to everyone. So maybe I'll refresh the, I'll put that in the discussion while you guys are presenting. I'll create an invite link. So many of you were on the ISO-Seq Learn Slack. That is an onboarding Slack. We have a permanent ISO-Seq Slack that is for people interested in continuing on. So I will send that invitation link through the discussion and also give you the link to the Google group. These are the 2 best places to continue to reach me and Kay. Other people at PacBio, Anna, Angeles, and Fran are also on these groups.
Hi.
So Fran, do you want to get ready?
Yes, I'm ready. Yes.
Well, we have 2 more minutes, and let me go do a bathroom break. I will be right back.
Okay, so basically with the previous part of the tutorial, we show the first scripts to go from reads to transcripts. Okay, so to transcript models in which all the reads that belong to the same transcripts are collapsed and presented. And then now with the next presentation that describes SCANT3, we will show how to annotate this data and to do the quality control of the data.
So I'm ready. So whenever it's fine for you, I will start. Oh, one more minute, just in case. Okay, so I guess I can start my presentation. So hi everyone, my name is Francisco, and I'm going to present my part of the tutorial, which is the transcriptome annotation and quality control using the tools developed in our lab, which are SCANT3 and ISOANOT. Once we have completed the ISO-Seq3 pipeline, we'll have a totally new transcriptome that will act as our own reference transcriptome. If you have several samples sequenced with long reads, we strongly recommend you to put them together and build a single de novo transcriptome that contains all the isoforms present in the different samples. Just like any reference transcriptome under construction, it must be compared with a priori information, remove possible artifacts, and annotate it. And in order to do that, without being Ensemble or NCBI, because we are not this type of organizations, we developed SCANT3 that will gather evidence around our new transcripts, and it will help us to discard suspicious transcripts and link them to already known genes and isoforms. Moreover, ISOANOT will assign different labels at the isoform level, like PFAM domains or microRNA binding sites, so we can load our functionally annotated transcriptome into tapas and complete the functional isotranscriptomics pipeline. Some of the questions that should be addressed before using a long read-defined transcriptome for further analysis are, for example, how similar are the isoforms compared to the reference transcriptome? Did we found already known isoforms, transcription starting sites or ending sites, or new splice junctions? What about those novel isoforms detected? Which are their differences regarding to the reference transcriptome? Are there any artifacts due to library preparation or sequencing issues? If we have complementary data, such as RNA-Seq from other samples, can we integrate all that information and obtain a better transcriptome? And a more tricky question, it's how can we describe novelty in a systematic manner? For that, SCANT will answer all those questions, but before going into details, I would like to show you the way of thinking behind SCANT with a true example. If we load into IGV the ISO-Seq3 defined transcriptome for melanoma cell line, which is publicly available, we can find this type of situation. In the reference, the gene STK10 only shows one isoform. Meanwhile, after sequencing with PacBio, we obtain 9 different isoforms coming from the same locus. Which one can be classified as known, and where are the differences? If we focus into the exon chain, 3 detected isoforms, these 3, exactly match every splice junction of the reference transcript. There is also one extra isoform, which is this one in yellow, that partially matches the exon chain of the reference. It's missing all these splice junctions, but it has all the splice junctions in the 5'. We can zoom in the 3' end of the transcripts, and regarding to the transcription terminated sites, we may question how similar the detected TTS are from the annotated one, but also if there is any poly-A motif close to each of the detected TTS. We did that and realized that the 3 upper isoforms, these ones, nail the annotated TTS and present a typical poly-A motif close to the end of the transcript. These motifs can also be found in the 3 isoforms at the bottom, but around 1.5 kilobases away from the annotated TSS. If now we zoom in the 5' end of the same isoforms, things get a bit more messed. We again can measure the distance from the reference transcription starting site to each of the detected TSS. And here you see a couple of examples of how the variability of the TSSs can go upstream and downstream the annotated goal. Someone may also question if we can use any complementary information about transcription starting sites in order to distinguish between true and false TSSs. So we thought about cage-peak data, which represent possible active transcription starting regions. And if we project these regions that are here, into the detected TSS, we can observe that some isoforms seems to be supported by cage data and can give us some clues about which isoform are most, let's say, trustful. But let's look into detail to the splice junctions. For example, all these ones. In this case, it seems clear that all the isoforms are reporting the very same splice junction found in the reference, this one. But we may also wonder if it has a canonical or a non-canonical splicing motif. For that, we look at the 2 first and last nucleotides of the introns, which are these ones. And we found this typical motif, GTAG, which is the most common canonical splicing motif. Imagine that we still don't trust this precise splice junction, even though I showed you that it is a known and a canonical splice junction. What type of additional data can we use to differ between a well-defined splice junction and a wrong one? If we have matching RNA-Seq data, we can map the short reads against the reference genome and see if this data supports the detected splice junction. In this case, as we can observe in the upper track, there are several reads that span perfectly the splice junction. This is a very, very nice example of a trustful splice junction. So now I'm going to show you an ugly one, and you will understand the problematics about the splice junction with this type of sequence. This splice junction, which is located at the 5' end and only present in the last isoform, is a novel splice junction. Moreover, it shows a non-canonical splicing motif, something which is very strange in nature. And on top of that, here, RNA-Seq data is not supporting at all the splicing event. I think that we will all agree that if we say that this splice junction is at least suspicious of being an actual splice junction occurring in our sample. We can also try to identify library preparation artifacts associated to long read sequencing, such as RT switching. So during the first strands, if you are not aware of this situation, during the first strand of cDNA synthesis, it is possible that the polymerase slips away if two homologous regions are close. In consequence, a fake splice junction can be detected when cDNA is sequenced. And what we do is to look at the genomic level for these homologous regions close to any detected splice junction. And if they are found, tag that splice junction as a candidate for an RT switching artifact. I'm sorry, but I couldn't find a really easy example for this type of problem, but I have one for intrapriming, which is also an issue associated to the library preparation. So do you remember those two shorter isoforms that were at the bottom of the ISCB caption? If we zoom in the 5' end of these two isoforms and look at the genomic sequence downstream, what can we find? A very rich genomic region of adenines that may have they may have interacted with the oligo-DT used for capturing messenger RNA. This may be the case of an intrapriming issue, and that's why the final transcript model seems to be shorter. We cannot assure you that this is true, but it's pretty clear that an oligo-DT may bind this region or a partially spliced transcript and obtain a truncated one. So once we are aware of all these possible problems, we believe that it was needed a tool to describe and address all these quality issues associated to long-read sequencing, put together supplementary evidence around the detected isoforms. We need something to help us to filter out those isoforms, suspicion of being an artifact. And beyond that, we will need to establish a classification system for novel isoforms regarding to the reference transcriptome. And I'm going to explain you this classification system because it will be very important to understand how scanting works. So for transcripts coming from known genes, we will classify as full splice match all those isoforms that perfectly match all the splice junctions of an already reported transcript, which in this case here is the blue isoforms. It exactly has all the splice junction of the reference transcript. Isoforms will be labeled as incomplete splice match if they partially match the chain of splice junction of a known isoforms, like here in orange, which is missing this first splice junction. There can be novel isoforms built up using a new combination of already known splice sites, and we will call them novel in catalog isoforms. In this case, in the green isoform, we are the transcription machinery is using an already known donor site and an already known acceptor site, but they weren't observed previously in the reference transcriptome in this combination. So it is a totally new isoform. However, if an isoform shows a totally new splice site, it will be labeled as a novel not in catalog. Here is the case of the red isoform, which present a novel acceptor splicing acceptor site. Regarding to novel genes, this is a bit more complex, and we classify that as follows. If an isoform maps exactly within the boundaries of an intron, it will be called a genic intron, like here in purple. However, if it overlaps both exon and introns of an already known transcript, it will be categorized as genic genomic, which is something that is not really explanatory, but it's describing a piece of a fragment of RNA that is cannot be associated to a gene, but it's overlapping an already known gene. There are also some cases that an isoform seems to overlap a known gene, even reproducing the splice junction, but it maps to the opposite strand. This is what we will call an antisense isoform, and it can be indicating that the reference transcriptome was not properly defined at the beginning, or that we have a problem during the library preparation. For example, some antisense isoforms are associated with intrapriming problems. It can also occur that a detected isoform spans 2 genes that in the reference appear to be independent. These isoforms will be labeled as fusion, here in yellow. And finally, from my point of view, this is the most intuitive category, which is intergenic. That means that an isoform is located in an unannotated region between 2 genes, and this is possible. This is probably a new gene to be defined and to be characterized in an not so well-studied organism. As you may imagine, if you are sequencing Drosophila or mouse or human, these well-studied organisms, these categories will not be really common. We will have more of full splice matches, incomplete splice matches, novel in catalog, and novel not in catalog. Here you see the inputs that we provide to Scanty. They are the sequences of our long-read defined transcripts, a reference genome in a FASTA format, and also the reference transcriptome in a GTF format. Optionally, we can give Scanty supplementary information like the one shown before. For instance, cage data, known poly-A motifs, or RNA-Seq related evidence, and Scanty will assess the degree of quality and novelty of each detected isoform. It will produce 2 files, a lot of files, but the main output will be a classification file, a junction file, and a PDF report. The classification file, it is a table. It is a TSV file with information at the isoform level. The junction file on the other side will contain all the information collected about each splice junction detected. If 2 splice junctions are shared by the same isoform, they will be reported twice because they are 2 splice junctions for 2 different isoforms. Using both files, these 2 files, Scanty will run an R script to return a PDF report with more general plots to visualize the characteristics of the new transcriptome. But it will also evaluate several attributes at the structural category level. As I said before, all these insights are important in order to curate our transcriptome. This curation can be done in several ways. One option is to use a machine learning approach, like the one we applied with the first version of Scanty. This is the 3rd version of Scanty, the last one, and the one we recommend to use. So far, we don't have an adaptive version of the machine learning approach, but it will be released soon. We have a second option for filtering, which is we call it filter by rules. And it is already part of the Scanty 3 tool. And we will talk about it later on the hands-on section of the tutorials. But of course, once you have a classification of the junction file, it is always possible to play around with the Scanty 3 outputs and decide your own criteria to include or exclude an isoform in your final transcriptome. Let's say that you don't want to keep all those transcripts that are shorter than half a kilobase. You can remove them from the classification, and it will be easier for you to filter once you have all the information of the isoform within a table. So now let's talk about the second tool that I will present, which is IsoAnnote. It is a framework for functional annotation at the isoform level. That means that it is a collection of scripts that needs to be adapted for each organism study in order to obtain a dense functionally annotated transcriptome compatible with tapas, our last tool that Angeles will explain later. We have already annotated, using this strategy, a bunch of reference transcriptome, and as you may expect, they are transcriptomes of model organisms, such as Arabidopsis, mouse, Drosophila, and human. This work was mainly made by our former colleagues, Lorena and Alberto. And now we are trying to put all this script together and make it as automated as possible, regardless of the organism chosen. For that, we are developing a snake-make pipeline that should work as follows. and to annotate reference transcriptome. First, we will download the annotation from public databases like Ensemble, RefSeq, and so. Then we do some formatting to prepare those files. And all that information will serve as input to run several annotation algorithms. Some will predict functional properties based on the transcript sequences, such as interpro-scan or UTR-scan. But there will be other algorithms that will transfer functional labels using information available in databases like UniProt. This will result in a series of we call layers here, and that will be easier to integrate. And finally, one last script will put together all these layers and will build a GFF3 file with the exact format that TAPAS is expecting. This is really cool, but the bad news are that right now ISOAnnote is very tedious and it requires a lot of computational skills and resources. So if you are interested in annotating your non-model organisms, we can talk afterwards. And we're working on finishing ISOAnnote, but in the meantime, we came up with a solution that is ISOAnnote Lite. We can take advantage of those already annotated, functionally annotated transcriptome, like mouse, Drosophila, and Arabidopsis, and transfer their features to the long-read defined transcripts. And of course, it will return a GFF3 file compatible with TAPAS. The good news is that ISOAnnote Lite is already implemented as part of Scanty, and it's as easy as activating a couple of options of Scanty 3, and it will go straightforward with the functional annotation. So I don't know if we are on time or not, but we can start with the hands-on part of the Scanty 3 tutorial. Do you have any questions? If there are any questions?
Look at the time. So no, you have plenty of time. You have 40 minutes until 6:50. So 40 minutes for you to go with the hands-on. Okay.
So for the hands-on section, I thought about 3 exercises that for using Scanty 3 and learn how this works and how we can interpret the results. First, we will perform a quality control for a subset of isoforms of PacBio's melanoma dataset. We will be using just chromosome 15 isoforms to make it faster. Then we have an exercise for running the Scanty 3 rules filter, which is really fast. And we will curate our obtained transcriptome and we can compare the results of the new report. And finally, we can also go for Scanty 3 quality control, but now activating ISOAnnote Lite options. And you will observe how different is the output. At least it will include a couple of files more at the output that will be used for TAPAS. If you want to put together the Scanty 3, this ISOAnnote Lite part with the first exercise, you're free to do that. It's really easy. It's just activate a couple of options that are already in the help of Scanty 3. But before running Scanty, please build this part you have probably done. If you were able to complete the installation part of the Scanty 3, you will have to build a conda environment with all the dependencies for Scanty 3. You will have to download GTF to GenPred, which is it's a side of this conda environment, and install CDNA Cupcake, which is already a prerequisite for ISO-Seq 3, I guess, because yes, for the collapsing steps that this is mandatory to have it. If you need more instruction, you can visit the GitHub of Conesa Lab with the Scanty 3. And you will also find Scanty 1, the first version. So you can see how things change and how Scanty 3 is now way faster with a lot of more categories. So, okay, the first exercise is just to run Scanty. So we'll have to activate Scanty 3 environment. I will go set the Python paths. And once we have the data, we run this command. You can copy paste it from the here, from the instruction part, that the installation part that you already have in Dropbox. And I have all this here. This is where my the exercise starts. Is if you go copy pasting, it will go smoothly. You log into your Amazon Web Services, and in my case, I'm in another type of cluster from the university, but it's the same. So first we activate the Scanty3 environment.
Fran, can you maybe make this screen a little bit bigger? That is easier to see.
Yes.
Yeah, thank you.
So I don't know if I can make the letter bigger, but what it's. So you have installed CDNA Cupcake previously, so you will have it in at some point in your directory or in some directory. In my case, it is exactly where am I. So the path will be set to here. Sorry. It's longer, but we can do that. It will be easy to copy paste all of this for you if you have the text file. And here, CDNA sequence. Remember, you have to both export the Python paths of CDNA sequence and CDNA Cupcake, both of them, because otherwise it won't work. Now I will just remove this part. Once this is done, and you have downloaded the data, I already have it on me on my working directory. I can export it. As you observe, I have a backup chromosome 15 because just in case everything goes wrong and I have a problem bigger than that, I have everything already run, so we can go faster if we run out of time. So if we move into this folder, you will observe all the files that we will use. We can also set some symbolic links to Scanty3. That in my case, as it is in the previous, in the upper folder, it will be easy to do this and to do Scanty3. No, sorry for this. My fault is this. No. How can I change a symbolic link? Sorry. I will try to remove this. Oh, okay. Can I remove this symbolic link without removing this? I'm afraid of this. Removing Scanty3.
Yes, you need to remove
the symbolic link.
File.
Okay.
And you want to remove the script.
Okay. Yes. I just now we have the symbolic links, and if you copy paste this part, we can start running Scanty, and I will explain in a minute all these different type of files that we will be using. So once it is working, you will observe some warnings. Don't worry about that. Okay, I have here some explanation of the different. Okay, you will observe that it will start running and predicting ORs and all these calculations, doing the mapping necessary. So I will explain all the different inputs. of this long command that I just copy pasted. First, we have the first input that we will need to give to scanty. In this order will be the FASTA file of the long read defined transcript. Then, and of course, the script, that's obvious. We will give it the reference transcriptome in the GTF format and a reference genome. We don't need an index in this case because we are using Minimap, which it won't need to build any type of index. Scanty one, and in first instance, it used GMAP. So if you are going to use, and you prefer to use GMAP instead of Minimap, which is something that I guess that you will have a lot of time to run that because it will take a lot, way longer. You will have to provide the genome index previously created with GMAP. In the case that you want to go straightforward and use Minimap, which is a pretty good option, you don't need to provide any type of index. You can provide scanty also the output prefix and directory to point where you want to store your output, and also the full length counts, which is the abundance txt file obtained after running ISO-Seq pipeline. This is how many full length counts are associated to a certain isoform that you are detecting, a certain sequence. Then we can also provide an expression txt file, which is a short read quantification of the long read defined transcriptome. So using RNA-Seq matching data, we can use Callisto and use the abundance TSV file. Remember, always using as the transcriptome that we are quantifying the long read defined one, not the reference, the long read defined one, because it will use the same IDs and all that information that is the one we want to obtain. We can also use RSIM to quantify, and wherever you use, we can also provide a scanty and expression matrix in which each of the rows will be an isoform, and each of the columns will be the different samples or replicates that you are using. If you have several samples, you can also use Callisto and RSIM and provide them as a comma-separated sequence of files, of Callisto abundance file or isoform results files from RSIM. If you have matching RNA-Seq data, and as I said before, we can use this information for knowing how good are supported the splice junctions by this RNA-Seq data, which is more precise to define a splice junction. We normally do that. We do that with using a star, and normally we do that without providing previously to star any type of a priori information, such as the annotated transcriptome. We just give a star the short read data, the reference genome, try to map and detect a splice junction. You will obtain a sj.out.5, and as it is, you give it to scanty. We can also provide CagePeak data from Phantom5, and also a poly A motif list of poly A motif that you can obtain from literature. Finally, the number of threads that you can, if you want to parallelize the job. Now let's see if this worked. It's ended. Yes, scanty just finished. You will see a lot of warnings and these kind of things. Don't worry about this kind of warnings, because it's trying to match all the information that it was given, and as it was split into 4 threads, it is warning you that some of the threads are have information from the other threads, and it's not finding all the correlation that it wanted. But don't worry, it finished. The scanty 3, this is a typo, scanty 3 report was successfully generated, and we can observe it.
Fran?
I think, yes.
Maybe before going into the PDF, since I think we have some time, maybe it's good that you show the isoform classification file that we just created, and then you explain a little bit the different features in the columns, because in this classification file, we have all the data, all the information that we extracted, and maybe you can go through that to show exactly which information is present.
Yes, of course. So here is the scanty3out folder that it was just created by Scanty, and this is the output. We have a TXT file with the params, that's so you can always keep track of everything that you are doing if you are running several times Scanty. So, okay, this is the input, the reference transcriptome, blah, 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 the expression file that you use, and so on. And what, okay, the first thing Scanty will do is to map the sequences. It will obtain a corrected DTF. We call it corrected because now if there was any type of a snip into that sequences of in the FASTA, we will keep the sequence in the reference genome. With this GTF, we obtain a FASTA, and we can predict ORF and obtain the amino acid sequences. And what Anna was talking about was about this classification file, which is a huge table, I think with more than 40 columns that you will observe now here, in which, okay, here is just one, for example, one row. For these isoforms called PB3.2, it mapped to the chromosome 15 in the positive strand. This is the length, the number of exons that were detected, the category, novel not in catalog, which means that it will present at least one novel splice junction, and the gene that it was associated to. Of course, as it is a novel isoform, we weren't able to associate it to a known transcript, so it will report it as novel. But if we find a full splice match, let's say a full splice match isoform, you will observe, ooh, this is crazy. Here, this is a full splice match. We have the gene associated and the transcript associated to with this full splice match. We also report the length of the reference transcript, which is this one, the number of exons of this reference, the distance to the TSS, to the TTS, the distance to the closest TSS or TTS in the same gene. So if one gene has several TSS or TTS because it presents several isoforms, Scanty will report which is the closer one and how far it is from your isoform. We have a lot of columns, like subcategories. If it has a RT switching states, it will be probably these false. If they are all the splice junction are canonical or not canonical, here in the first, here in this first isoform that we were talking at the beginning, it presented all the splice junction canonical. The minimum number of samples in which this splice junction were found. This is a lot of information, and all these different columns are described in the Scanty3 GitHub. If you have more questions about them, but also, like if it is within a big cage, it will be very useful having this table to filter out all those isoforms that we don't like to, and also to plot several things. Junction file works pretty similar, which is, where's the junctions? Here. So for this isoform, we have junction 1, 2, 3. Then for this isoform, junction 1, 2, 3, 4, 5, 6. We have the genomic coordinates of this splice junction. If it is a novel or a known splice junction, but also if it is a known or novel donor or acceptor site, distance to the closest known and donor acceptor site, the motif that is the splicing motif found in the intron, and as I saw before in the presentation, if it is a canonical one or not. We accept as canonical 3 different types of motifs. but this is the most common one, GTAG. And well, of course, if it has support, short read support, how many, which is the total coverage, which is here, the total coverage will be 16 reads mapping this splice junction. And so on, this is a novel splice junction with 7 reads spanning this precise splice junction, which is some clue about that the novelty can be actually there. And all that information can be used to generate plots and to have a better picture of how is the transcriptome we are handling. And it will answer most of the main question that we may have about our transcriptome, like how many isoforms are already known. So for instance, this is the first page that you will obtain in your ScanT3 report. It will tell you, okay, we found for this little subset of the chromosome 15 more than 400 unique genes and more than 2,000 isoforms. We classify these genes between annotated and novel ones. And here you will observe the different categories that I explained before. And as expected, full splice matches were the bigger group. And of course, also the splice junction, if they are non-canonical, non-canonical, and so on. But how many isoforms per gene were found? This is really interesting if we are assessing the degree of splicing that is occurring in our samples. We can say, okay, for isoforms per gene, so more than 30% of the genes that we detected presented more than 6 isoforms, which is a lot. But also, it is different if we are talking about annotated genes or novel genes. So annotated genes are more prone to have several isoforms. Meanwhile, the novel genes that were detected mainly have just one isoform. So this is more likely that these novel genes are not actually, they may be noise. So one of the questions also we are sequencing with long reads. How long are these transcripts that we are detecting? So we provide also this type of plots, which in this case is not really smooth because we just have the isoforms from chromosome 15. If you have a larger, you use the whole transcriptome from this very same sample, you will have a really nice histogram with almost no peaks at all. We can also wonder about the detected TSS and TTS, because now we are very interested in them to actually know how full, if the full transcript that we are getting are actually full length transcripts. So full length, full splice match isoforms are capturing known TSSs or TTSs. So yes, this is about more, bit less than, it's about 30% of the isoforms are, because I know the percentages, are actually nailing it or really close to the TTS, but the TTSs are, we are detecting some other different TTS upstream and downstream the annotated ones. However, for the TSS, as you may know, that it is sometimes it gets complicated to get to the 5' end of the read and of the transcript and get a truly full length transcript. And sometimes we don't. We get this slope in which this center part will be the annotated TSS, and this will be closer TSS detected. But as you will see, most of them have cage support. So maybe they are actually true alternative transcription starting sites. The main question of all of this is if we can trust the isoform, all the isoforms described. So for novel, we plot also in the PDF report, this what we call quality control attributes, and we distinguish between structural categories because it's not the same to find a known isoform that was reported previously in several occasions and it's present in a curated transcriptome, like in gene code, or finding a new isoform with a novel splice junction. So as you observe here, novel not in catalog isoforms are enriched for what we call bad quality attributes such as splice junction without coverage, to present non-canonical splice junction, something that is really rare in full splice matches, in complete splice matches. And also they are increased for predicted non-mediated decay, which is a signature of maybe a wrong isoform. So I guess that I think that, if I'm not wrong, in the Scanty report you will find almost 80 plots. So there are a lot of ways of observing the same data from different point of view. If you have questions about them, I will be happy to answer all of them and to explain any plot that you will need. I cannot go through the 80 different plots in this presentation because I would like to go for the filtering. So now we have a transcriptome which has been classified and it has been evaluated. But as I saw here we have some isoforms that we don't trust them because they may be noise and they can produce a lot of problems downstream in our analysis. So we'll need to remove those possible artifacts. And using the Scanty3 rules filter, full splice match is isoform. The rules are as follows. Full splice match isoform will pass the filter unless they are cataloged as a product of intrapriming, which means that downstream the TTS, we find in the genomic sequence more than 60% of adenines. That's what it stands for, this -8 0.6. We can change this parameter. If you are not so confident with 0.6, you can say, okay, 0.4 or 0.8, that's your call. And for the rest of the categories, an isoform will be discarded because of 3 reasons. Because of intrapriming below this threshold, because it was tagged of having one splice junction with RT switching, with an RT switching artifact, or because it presents a non-canonical splice junction without short-reach coverage. We call an splice junction to have no short-reach if it is below a certain threshold that we can change, because it will change a lot if we have a sample really deep sequence, or we have a really shallow sequence, and maybe if you find 2 short reads spanning one splice junction, you may trust it. If you sequence it a lot, you may increase this number, so you are more stringent filter. And we provide just 3 files: the corrected FASTA that it was created previously, the corrected GTF, and the classification file. That's all that the Scanty3 filter needs. Let's go for that to run it. Here is this Python 3. Oh, where am I? Okay, I changed the directory, so I go back and Scanty3 output classification, the corrected file. It will go. This filtering will do everything for us, not only removing all those artifacts, but it will also create a new classification file, a new PDF report, so you can compare both outputs. And it will also report, I think that it will report you why each of the isoforms were discarded. And also have a filtered GTF. So if once you have filtered your transcriptome, you want to continue and perform, let's say, differential expression analysis, you can quantify with that filtered transcriptome. So once this script is running, I will show you how this changed. Now, instead of having 400 genes, we have 370 genes, so about 40 genes were removed, and about 300 isoforms were excluded and removed because they were tagged as artifacts. Most of the novel non-canonical junctions were removed. Now we just keep 11 splice junctions because they have a higher, they have a coverage over 3 short reads. And now you see that the parameters compared to the these quality control attributes has improved in these 2 categories, especially in novel not in catalog. Now arti-switching artifacts are almost inexistent because that was one of our rules, that no arti-switching is allowed between any category except for full splice matches. This is more or less easy, and this works probably. Well, it will take a bit, but there's nothing really different compared to the other output. It's the same, but filtered. And now we can use isoanode option. How much time do we have?
10 minutes.
10 minutes.
Oh.
Okay. So isoanode options are these two options. And they will create your own tapas compatible GFF3 file with functional annotation at the isoform level. So there are a few things to have in mind. First of all, the reference transcriptome that we are using should match with the one that we are providing for this copy paste that isoanode does. So we have a reference transcriptome annotated, let's say ensemble transcriptome annotated functionally with all these labels at the isoform level. What we do is kind of a copy paste, and we pass these labels if they present the same coordinates, if it is providing, if an isoform is providing the same coding region, we can assume that the same PFAM domain will be observed in the protein because it will be the same. If a different UTR motif, if a certain UTR motif is present in an isoform, in a 3' end of an isoform, can be passed to a similar one, even if the exon chain in the middle of the isoform is different. But we need to keep the same IDs. That's why these two reference transcriptome and the annotation file must be matching. We have the isoanode light option. If we don't have any information, let's say we are working with a snail or any type of animal that is, or organism that is it's not being annotated yet, we can go to tapas, but we won't be able to use functional annotations. We can activate this isoanode light option. It will create the GFF3 file, but without information about these functional labels. That's the bad part, but with tapas, you will be able to perform any type of differential expression analysis, differential isoform usage, a lot of analysis are available, will be available in tapas, but not all of them, because we will be lacking of information. If we activate the GFF3 option, we have to provide this file, and you can find all these GFF files in here in the tapas webpage. And as I said before, we have information for human, mouse, Drosophila, maize, Arabidopsis, and I don't know if I'm missing someone.
Mouse on human.
Yes, mouse on human from RefSec and Ensemble. That's it. I think that if it's not ended, yes, we ended the report. So now I can show you the output. So here now we have all these files that are totally new, which are the filtered light junctions, FASTA classification. Now everything is filtered, and the reasons that I think that it will be the most interesting part. Well, okay. I will show you more. These are the reasons. For example, this certain isoform was removed because intraprimate, because a low, this one because a low coverage and non-canonical motifs. So you will have a description of why you are taking out some of the isoform, and it will be easier for you to justify your filtering. And if you have any type of problem or issue during the library preparation, you will observe these type of things in these reports. And okay, we were talking, I have like 5 minutes, right?
Mhm.
So I'm not running the last part of so this part of the code is just like before, adding these 2 options. I would like to show you how the output changes. So before, after running scanty, we obtain this type of GTF. This is what we call corrected GTF, which is just the description of the transcripts and the different exons that one transcript has, with a transcript ID and a gene ID associated to it. Then, after running ISO-Seq 3 with isoanode light, this changed a lot. And we are including these functional labels like UTR sites, which is in this genomic location with a certain ID and some description, gene ontology, if cake pathways, because we also associate genes to certain cake pathways, PFAM domains. We have also annotated post-transcriptional modification, binding sites, and so on. And all this information is used by TAPAS to create these type of figures. This is the representation of what we do, a representation of a protein, in which we can find 3 different PFAM domains, which are these 3, that if you Google them, you will probably obtain the information about what is each of the parts. Several post-transcriptional modifications, and binding site here in blue, which is this one. And this is really useful. And we can, as Angeles will show you now in TAPAS, all this representation can also be used for analysis of differential inclusion of maybe one isoform is excluding this part, this signal, because it has an alternative TSS, which is really, it may be interesting for your studies.
Just a reminder, it is now 5 minutes till the coffee break.
Perfect.
So we'll wrap up in 5 minutes, give people like a 10-minute mental break. So then we can go into...
I'm not with presentation, so this was my last slide. I will now, this is just like the preview of how TAPAS will look like, and Angeles will explain that in a minute. If you see me look to the right, it's because Angeles is here. So that's it. If there is any question, I don't know if the chat is going on.
For now, there's nothing on the chat.
Okay, that's good. Then that means that everything was fine or everything is asleep. We have 2 options. So, yes, coffee break.
Yeah.
Yes.
Yes, if you have questions, please write them down and we will answer them.
The live Q&A session.
Yeah, we'll stop sharing. Okay. What happened if we don't use them? Okay, yes, the question of if we don't have a model organism. So functional annotations won't be available. You can use isoanode light and you will be able to load your transcriptome into TAPAS. But it will be easier. Now this figure will appear to you just like the black line. So you will have a predicted protein and a transcript, and you will be able to perform some of the analysis included into TAPAS, but some other that are related to functional annotations, like the differential feature inclusions. But maybe we are interested in the inclusion or exclusion of a certain nuclear localization signal, for instance. We won't be able to do those analyses. But with a couple of clicks, I think that it is interesting, especially for those that are not really used to perform differential splicing analysis and differential expression analysis. TAPAS, if you run isoanode light without anything, just the isoanode light option, without the GFF3 option, it will run. You will obtain a GFF3 file, and you can load it into Tapas. Tapas doesn't. It will be a bit not so funny to play around, but you will be able to do some things. Yes. And I guess that's for model organisms. We are trying to improve ISO to work with any type of model or non-model organism, but it's really tricky to put together all the different information from different sources and make that run. Because okay, once we think you are done with, okay, I'm able to annotate Drosophila and Drosophila simulants, and finally you go for the next organism and everything fails. And I don't know, it's a problem. We're working on that. More question? We have this. Do you use the number of supporting ISO-Seq to be calculated? Mm-hmm. Sorry, I will start, sorry. ¿Me salió? Ya no me salió.
So if there are not more questions, we can have a 10 minutes break. I think that's when we have the coffee break, so that you can refresh yourself and get ready to work with Tapas. And we will come back at the top of the hour. And we will be online in case you want, in the meantime, to ask any questions. So we are still here. So we will be back at 7 p.m.
So I think there are a couple of questions in the Q&A. I don't know if maybe Anna or Liz wanna answer them. I'm not sure I can.
So
Yeah.
I guess that to improve confidence on the identified novel transcript, I guess that is to, okay, if we find a novel isoform, how can we say, okay, we trust it more than the other? For that, I will recommend always to, if you sequence with ISO-Seq, to always have from the same samples, not from the same library preparation, but from the same sample, sequence also RNA, do RNA-Seq, a couple of replicates. That will help you to quantify better, because the dynamic range of RNA-Seq is bigger than that PacBio, so you will be able to quantify better. And it will also help you to, as I said, for splice junctions, if they have support from short read. There are two different technologies, and if you are able to see something that is novel, a novel splice junction with two different technologies, that's more likely to actually be true, and you will be confident about having that. And that's my main recommendation to improve that confidence. And because that's novel transcript, that's also, if you have support from several points, from several sites, that will help you always. The fraction that you can annotate for a novel organism using ISO-Seq, I have no idea about if we mean about annotating to describe the genes that we are finding, it will depend a lot of the organism, I guess. It will be pretty different.
What is the question? I'm not seeing the question.
Mouse. The fraction will be, I don't know.
What is the question? Because I'm not seeing the question.
To detect all the expressed genes. Of course, you won't be able to detect anything that is not being expressed. But the rest of the genes and the isoform, probably the genes for sure. The isoforms may be a bit more tricky because you will be mixing some things. But the genes, I guess that technically everything that you are capturing with the poly-A, you will be able to annotate it. Then it will be the difficult task to describe a function for that and for the different part of the isoform. And
so the questions are in the
differences in quantification between RNA-Seq and ISO-Seq. Yes, there are pretty much differences. If you quantify with ISO-Seq, you will be limited to the number of reads that you are having, because if you are sequencing with, let's say, really deep, I don't know how deep you can sequence with ISO-Seq, but right now with the newest platform, but these differences, I guess the dynamic range, if they're really low expressed transcripts, you won't be able to detect them probably with ISO-Seq. And with short read data, you will be able to capture evidence about those transcripts that are there. But you won't be able to reconstruct the entire sequence. And it will be also a problem, the dynamic range between these two different types of sequencing.
Can you hear me?
If you observe an increase of 50% of the number of reads associated to a transcript using long reads, maybe that's an increase from 4 to 6. Meanwhile, if you are using short reads, that 50% will be a broader range, and you will be more confident of that this 50% of increase is actually happening. It's not just by chance. You're capturing a couple of more molecules, and you get this output of a differentially spliced or differentially expressed gene, which is actually not. If you want to add anything, Anna?
Yes, can you hear me?
Yes.
We cannot hear you.
Okay, because I was trying to talk and you were listening.
¿Tú la escuchas? Okay.
Okay, so what we have seen about the quantification is that with the new SQL2, and I think that Liz also knows better about this, is that you will have a better quantification only using long reads when you are in the middle and high range of expression levels. So for those transcripts that are medium to highly expressed. For those that are more lowly expressed, the quantification will be noisier. I don't think that so far there has been an exhaustive analysis on this. In our lab, we are trying to get some kind of light about these dynamics, but this is roughly what you would expect. Right?
I think we have 3 minutes until the last session.
Mm-hmm.
So on the ECCB discussion, I am going to paste an invite link to the permanent ISCB Slack channel. And this link is valid for 30 days.
Okay, so Christopher is also asking which are the differences in quantification that we see between RNA-Seq and ISO-Seq data. So in the Scanty paper, we describe a little bit about this. And one of the things that happens is if you don't have ISO-Seq data and you are going to be quantifying your transcripts only based on short reads, mapping to a reference transcriptome, sometimes you are going to be quantifying the isoform that is actually not expressed, because this level of novelty that we see every time that we sequence with long reads. So in this case, there will be a few isoforms that are simply wrongly quantified if you are only using short read data, just because the true isoform is not in the reference. This is if you compare going like the short reads RNA-Seq versus the going the ISO-Seq RNA-Seq. If you are just saying, okay, I create my transcripts with ISO-Seq and then I look at the expression level from the long reads from the matching short reads, I don't think that there is a clear pattern there. It's simply that the low expressed transcripts will be more accurately quantified with the short reads data. I hope that I answered that question. The difference on the gene expression level are very minor, I would say. And again, this is for the lowly expressed genes. Liz, Angeles?
Yeah, should I start?
Yes.
Cool. I'm gonna share my screen now. Is everybody seeing my presentation?
Yes.
Cool. So I'm gonna put it on full screen. Here. Okay. So hi everyone, my name is Angeles, and well, we're gonna move on to how to do functional isotranscriptomics with tablets. I hope everybody could install the tool. It requires a couple of dependencies, but if you have Java and R and you haven't had the chance to install it, you might as well just go to Tapa's GitHub and try and download the jar file. Try and see if that works, and that way you can follow the hands-on. Otherwise, just sit back and relax and watch. So to better understand the aim of TAPAS, I'm going to first explain what functional isotranscriptomics analysis is. Traditional isoform expression analysis evaluates differential expression of the different isoforms of a gene between conditions, and it can provide this type of information about changes in the major isoform expressed in each of them, the conditions. However, this approach presents several limitations. First of all, it is not easy to systematically visualize the differences between isoforms that have been generated due to splicing. And in addition, the biological readout of these expression analysis is difficult to interpret, mostly because there is no way to know how much the isoforms of a gene actually differ in biological function. So to make these two tasks easier for users, our group has designed some strategies to incorporate functional elements into transcriptome annotation using isoanode and isoanode light, as Fran described before. Then in TAPAS, what we do is provide analysis that combine expression estimates with these functional annotations and enable functional isotranscriptomics. Furthermore, and this is one of TAPA's coolest features, TAPAS provides interactive maps that we can visualize to see these functional features within the sequence, their precision, and so on, to understand the actual functional differences between isoforms. But before I dive into TAPA's functionalities, I'm just gonna show you a brief example of how this works. Imagine that we have this gene, the cadherin delta gene, and we, thanks to the functional annotation, instead of only knowing that there's a switch in isoform expression between these two conditions, we actually know the correspondence between these isoforms on the left and the different features and motifs and domains that they contain. This is at the protein level, for instance. So if we were to analyze one feature, take for instance this nuclear localization signal, we would, by combining the expression of feature including and feature excluding isoforms, TAPAS will be able to detect a change in feature inclusion that is totally condition dependent, with the NLS containing isoforms being favored in the oligodendrocyte condition and the feature excluding isoforms being just favored in the neural condition here on the left. So this is in fact a real example that we validated for the TAPAS paper and that you can find if you're interested in and read it. And this is the type of insight that we're looking for. I'm now gonna move on to discuss how TAPAS works and the inputs and how to create a project, and give you a brief tour of TAPAS interface and the main analysis functions before I move on to the hands-on. TAPAS works in the form of different projects, and projects are the main instance in which TAPAS is based. A TAPAS project will require 3 inputs: a design file, an isoform level expression matrix, and a functional annotation GFF3 file. And this can either be selected from those built-in TAPAS or provided by the user. The design file is a small text file specifying the names of the different samples and the conditions that they correspond to. In the case of this tutorial, we're going to use a data set that has 2 conditions, one neural precursors and the other oligodendrocytes, with 2 replicates per condition. This is very important because TAPAS requires at least 2 replicates per experimental group in order to work. And it's also very important that the sample names in the design file match the column names in your transcript expression matrix. So make sure that correspondence is there. Also, do not include an isoform ID column in your matrix because otherwise TAPAS won't be able to read it. However, 2-group comparisons are not only the only type of design that is currently supported by TAPAS. Other available designs are single time course, containing a single experimental group, but several time points with observations, and multiple time course with 2 experimental groups and the time-dependent design for each. In this case, you need to add the time column to the experimental design file. Finally, as I said before, TAPAS requires a functional annotation file that has a compatible GFF3 format. This corresponds to the output obtained after running scan-T3 with the isoanode light argument. So you don't really need to do anything else. But how, if I've done a long read experiment, how would I go about generating these inputs for TAPAS? Well, I have outlined here a brief example going from a long read experiment into those input files. This is just one possible way to do so, but this is a strategy that we routinely apply in the lab, even though there might be more than one valid strategy depending on the aim of your study. So let's imagine that I have a two condition experiment in which I have neural precursors and oligodendrocyte cells from mice and 3 replicates per condition. To obtain long reads, one could pull cDNA from all samples from the same condition and sequence using PacBio. To complement the information provided by long reads, we generate short reads for the same condition and for each condition and for each replicate. And this is very important because of what Fran said about quality control of transcripts, but also to do quantification downstream. Next, long reads for bug conditions should be pulled prior to processing in order to obtain a single transcriptome for our whole experiment using ISO-Seq3 and SCANDI3 as it has been shown in this tutorial. Ultimately, ISO-Seq and SCANDI3 will provide two outputs, a GFF3 file containing the functionally annotated isoforms and a filtered and curated GTF file. This is our experiment's transcriptome. Finally, short reads from each sample must be mapped to the genome using this GTF, and subsequent isoform level classification can be performed. And the output of this process will be the isoform expression matrix that we need in order to create a project in TAPAS. So how do I do that? How do I actually create a project in TAPAS? Well, you need to first run the application, either by double-clicking on the tapas.jar file or by running the java-jar command as shown in the slide. But before we proceed, it is very important to locate the input files that were made available before the tutorial, as we will need to provide them to TAPAS upon project creation. And if I'm not mistaken, this were available in one of Dropbox links that Liz provided before the tutorial. So after starting TAPAS, you will see this window and you need to click on the new project button. And a sub-window will open, and in this sub-window, users need to specify the following information. First, a name for the project. Then we need to supply an annotation file. To do this, we will first specify the species and then select whether we'll be using one of TAPAS built-in annotation files or providing our own. In this case, we must use the button down here to provide a path for our own annotation file. In the next section, we will indicate the type of experimental design that we're working with. And in our case here in the hands-on, we're gonna be working with a two-group comparison, but please keep in mind that we also have the two other designs that I described before. Then we need to supply the design file and the isoform expression matrix. And to correctly do this, you can always click on the help button down here, which will provide more information about the format if you have any doubts or don't remember how to build a design file, for instance. Once you have specified all of this information, two additional options for normalization and count filtering are available. For row counts, TAPAS provides the option to use TMM normalization. However, users are welcome to apply their own batch effect correction or normalization method prior to loading their data into TAPAS. And if you do that, you can just unclick this. However, keep in mind that you need to provide your expression as normalized or corrected read counts. We don't accept TPMs or CPMs or any other type of expression matrix. And in addition, locally expressed isoforms can be filtered just by defining a CPM cutoff value. Finally, users may provide an inclusion or exclusion list in order to select a subset of the isoforms for their new project without any need to manually filter the expression matrix or the annotation file. And this is very convenient when you just want to analyze a subset of your isoforms. So before I continue, I'm going to try and demonstrate how to create a project using the provided input files. And then I'm going to leave some time for you to create your own project. And since project creation is going to take a little bit, we'll leave TAPAS running in the background and come back to the presentation, and we will start the hands-on. And when we do that, it will be all ready. So you should have downloaded, oops, sorry about that. You should have downloaded the tapas.jar file. The most recent version is tapas 1.0.3, and in a folder you should have somewhere in your directories in your computer, you should have these 4 files, which are the ones I'm going to use to create a new project. So if I double click on the tapas.jar file, I will see tapas open. It's right here, and I have the new project button as I described before. So if you click on that, you'll need to enter the information I was just telling you about. First of all, we're going to be giving it a name to the project, for instance, ECCB tutorial. And then we're going to select the species that we're working with. In this case, we'll be working with a mouse dataset. And we will be specifying our own annotation feature. So we see what that's like. There are, of course, available reference annotations for mouse. We're going to provide our own EFF3 file, which is included in the folder I provided. So if I go to tapas _ project data, the first file that appears is this GFF3 file. This is the annotation that you need to provide. And if I click on open, I'll see the path appear down here. Of course, you can manually type the path too, if you need to. Down here, we're going to select the experiment type. As I said before, we're going to be working with a 2-group comparison design. And I'm going to provide the design file and the matrix. Just as I did before, if I go to my home directory and I go to tapas project data, I have the experimental design.tsv right here, which is the file that I need to provide. Same thing for the matrix, which is called input matrix.tsv. You provide that. And all we have left to do is provide a transcripts filter. I'm going to leave the normalized data and expression value filter selected because we really recommend doing both things. And then I'm going to provide an inclusion list. This is just a TSV file that contains transcript IDs. I'm going to show you. This just contains transcript IDs that I selected in order for the tutorial to run faster, because the data set I provided is just a really big one. So I just selected a subset of transcripts that would be interesting to analyze. It's about 2,000 isoforms. So in the same folder, I have the transcripts, example transcripts.tsv file. I'm going to provide that to tapas. And once you've done that, you'll be ready to go. You just need to click on OK, and you should see the project start creating. I think I'm going to leave you 2 to 3 minutes to do that yourselves. And I will go back to the presentation to continue. I'm just going to leave my new project window open like that in case you need to look at it for reference. And in a couple of minutes, I will be back to the presentation. If in the meantime, if you have any questions, you can just type them.
So there is a question about how you link your RNA-Seq quantification to the ISO-Seq to the ISO-Seq transcript model. So basically, the idea here is that once you have created your reference transcriptome with long reads, and then you have curated it and filtered it with scanty3, then you have a set of transcripts that you trust. And these are the set of transcripts that are expressed in your experiment. So you take this as your reference transcriptome for mapping your short read data against. So instead of using like the reference, that is normally what people do, you use that one transcriptome that you just generated, and then you quantify with the short reads. I hope that's clear.
Mm.
But you can have several isoforms. Yes, you can have several isoforms of the same gene. So, but you must use a mapper that is able to differentiate between isoforms. For example, our sim of Callisto will do the job. The advantage of using the express transcriptome is that the complexity of your transcript is much more reduced in comparison to the reference transcriptome. So if, for example, in human, you can have up to, I don't know, 200,000 transcripts annotated in the reference, in the global reference. When you are analyzing a particular tissue, maybe you have expression of, I don't know, 20, 30,000 transcripts. So the complexity is much easier. So when you go with your short reads against that simplified transcriptome, it's much more accurate to resolve the quantification of the transcripts. Yes, you limit the number of transcript models because you detect the true transcript models that are expressed in your experiment, in your samples. That's the idea. Plus that you also identify those that are not in the reference, and that's something that we have seen that happens a lot. So you usually find many novel in catalog transcripts, so it's a new combination of existing exons that are not annotated in the reference. We know that the current, even in the human and mouse and model organism references, are annotated for this type of combinations. So by sequencing your samples, then you know exactly which transcripts are being expressed.
So I just added a question to the poll asking whether you managed to create a TAPAS project in order to just have an idea of whether people were just able to do that and if you will be able to proceed with the hands-on. If you got an error, you're welcome to ask perhaps later, because it's kind of hard to troubleshoot installation stuff, but we'll be happy to help afterwards via the Slack channel. For now, I'm gonna continue with the presentation. I was just about to give you a brief tour of TAPAS interface. Okay, because one of the most interesting features of TAPAS is that we actually have a GUI that we can interact with, and this makes it really accessible for people that don't have a computational background. So after you create a project, you should see TAPAS main window, which consists in 3 main areas. First, the control panel at the upper bar, the data panel at the top, and the visualization panel at the bottom. The control panel contains 3 main areas. First, the projects button, which provides access to extant projects and the option to create or delete one. A series of analysis buttons, each of which contains several analyses associated to each of TAPAS analysis modules, which I will describe in a minute. And here you can find the results for already run analysis that you may have done before. And a search bar to filter the data and result tables shown in the upper panel. When users select to run an analysis, this type of window will pop up. And in there will be analysis specific options to set parameters, or maybe select functional categories to analyze, or provide a name to store the analysis results. In addition, each window has its own help button. So just keep an eye on that and bear in mind that while you're using TAPAS, there will always be more information about the analysis parameters and their interpretation. The data panel is designed to show tables, either expression matrices for different features, which are genes, transcripts, and CDS, or result tables for the different analysis, all of which can be opened simultaneously in different subtabs, as you can see up there. Of note, TAPAS can open up to 3 projects simultaneously, each of which will show as a new main tab in a new color code. The data panel has also a multi-option bar on the left-hand side, and this bar provides options to view the log of the analysis, filter the table, export the table, access plots, rerun the analysis, and also it has a help button that provides info about the variables included in the column. And on the right-hand side here, you have a plus button that enables you to either add or remove columns from the table. If you click on the graphics button on the multi-option panel on the left-hand side, you can access the specific visualization options, and graphics will be shown on the visualization panel. Different visualization tabs will appear for each analysis graphics, and when gene level visualization is selected, several sub-tabs showing different layers of information about a selected gene can be accessed within that main tab. For instance, the transcript annotation, the predicted protein annotation, and also the expression charts. In addition, the visualization panel also has a sidebar on the left-hand side that contains graphic-specific options, including viewing the log for the plot or exporting the images, and also customizing the visualization. Finally, there are several ways in which you can access built-in documentation within TAPAS. It is mainly using the help buttons that are available all through, but you will be able to learn more about plots, tables, and analysis, and this will greatly aid the interpretation of results. So before I go to the hands-on, I'm going to describe TAPAS analysis modules real quick. Although the tool includes several analyses, these can be divided into 3 modules, depending on the aim or the type of insight that they provide. First, the functional diversity module compares all isoforms for each gene in order to provide qualitative evaluations of the diversity of functional labels in the transcriptome. The FDA module therefore works as a tool to assess the level of variation that is generated in the transcriptome due to splicing and the functional categories for which variation is greater, but it does not take expression into account. In turn, the differential module evaluates expression changes for the 3 feature types supported by TAPAs, that is genes, transcripts, and CDS or predicted protein. The differential module uses traditional differential expression analysis, but also assesses changes in the isoform expression within a gene via the differential isoform usage analysis. DIU can be run considering transcript or protein isoforms, and very importantly, all of these results can be very easily coupled with different types of functional enrichment analysis in order to assist biological interpretation. Bear in mind that TAPAS supports enrichment for Go terms and pathways, as is the traditional way to do enrichment, but also for the rest of functional categories that you annotated in your GFF3 file. And this is very useful when coupled with DE and DIU analysis, because this kind of helps you figure out which functional features are the most commonly present in genes regulated by splicing and give you some clues about the role of this regulation. And finally, the feature analysis module combines isoform level expression and functional annotations to estimate rates of inclusion and usage of functional elements across different conditions or time points. These analyses constitute the core of the functional isotranscriptomics and provide direct readouts on the functional consequences of alternative isoform expression. TAPAS, of course, supports this type of analysis for all functional categories in the annotation, as well as poly-A sites and global changes in UTR length. So now we're gonna move on to the hands-on session. In my slides here, I included a lot of exercises that you're welcome to go through on your own if you want to, but we only have half an hour left. So what I'm gonna do is I'm going to run a selection of analysis to give you a feeling of how the application works, and you're welcome to follow me on your own computer. I really encourage you to use these exercises as a guide to be able to figure out what kind of analysis you can run on the application. I'm going to first open the project that I created before the tutorial, and it will show me the main panel, which has all the sections I described before. So we have here the 3 different modules: diversity, differential, and features. And also enrichment is separated, but this is closely related with the differential module. First thing I'm gonna do, imagine that I'm going into this data set and I don't know anything about it. I might wanna know how much stuff have I annotated and how many isoforms within a gene lack or have different features, right? So I'm going to do that by running a functional diversity analysis. Functional diversity can be run either at the category or at the ID level. So the difference, as you're going to see in a minute, is that if I analyze them by category, I'm going to have a gene-wise result. For instance, let's say I'm going to call this FDA all because I'm going to analyze every single category. And I can do this using 2 different criteria, genomic position or feature presence. If I use genomic position, the program will label my feature as varying within a gene if within isoforms there's a change in the positions that define this feature. Whereas if I use presence, the variation is going to occur only if one of the isoforms within the gene lacks completely the feature that is annotated. It is very simple. There are categories within some of these that you're welcome to explore on your own. I'm going to just click OK and run it. In case you will see a log appear and the table will show once the analysis has finished. Luckily, I already have my results. I call them FDA posts because I used position as a criteria. And this is the type of results that you get on the FDA category analysis. It's a gene-wise table. And for each global functional category, for instance, repeat regions or 5' UTR motifs or UORFs, I have varying or not varying depending on what the analysis found. And I can rank the table by any of these and add new columns and do whatever, play a lot around with this results table. The one thing that I can do is obtain a summary visualization of the results by clicking on this button here. And this will show me a plot down here that summarizes all of this information. Instead of having it on a gene-wise manner, I have it by category, and I can see on the x-axis the percentage of genes that have a feature. For instance, I have 3' UTR length, which of course is very widely annotated because most genes and most isoforms have a 3' UTR and have a given length for that. There are other categories that a very low percentage of genes have, for instance, intramembrane domains. And this is what it gives you. And the shaded area in the bar will indicate the percentage of variation for its categories. For instance, if I go to the 3' UTR motifs and I leave my mouse here, I will see this little window appear where I have the percentage of genes that have this feature that show variation for it, right? So it's kind of a nice summary of the whole FDA analysis. And here on the right-hand side, I have the same, only it's pairwise. This means that instead of taking a gene and comparing every isoform within, and if just one isoform is varying, I label it as variant and I count it once. Here what I do is within each gene, compare every isoform to every other isoform in a pairwise manner. I do as many pairwise comparisons as I can, and I report the percentage of comparisons that showed variation. So the higher, the more isoforms within a gene have variation. Whereas here, just by one isoform being varying, the whole gene will be labeled as varying. It's kind of an interesting exercise to compare the genomic position FDA results to the presence-absence results. I already have run them. I'm not gonna go through this much, but if I close this and I reopen, I'll be able, and you'll see how in this case, when it comes to presence-absence, it's a much more stringent category criteria for variation. So there's lot less variation in proportion. This is the FDA by category, but there's another option to run FDA by ID. And what this means is that I will only be able to select one category. For instance, let's say I'm interested in PFAM protein domains. I'm going to select this that. I'm going to label this ID PFAM2 because I already run it. And I can do the same. I can change the criteria. I'm going to keep it as position, and I'm going to run this, and it will show the very quick. And as you can see in this case, I have only selected one category. Therefore, I am analyzing every individual PFAM ID within the PFAM category, and I'm counting the number of genes for which this domain ID is varying. For instance, I have protein kinases, and I see that for all the annotated genes with a PFAM domain, there's variation between the isoforms for 12, and 7 of them are not varying. I can actually right-click on this line and select drill down data to see a full list of these genes. These are the genes that are not varying, and these are the genes that are varying. And I would really like to show you an example. For instance, CLK4, if I right-click on it and I click on Show Gene Data Visualization, I will be able to see one of the cool maps that I was describing in the presentation. These are graphical maps for all the isoforms where you can see the differences between them. These are the functionality features, and this is an interactive map. So you can scroll over and see what everything is, and there's a legend down here. If I go to the protein, which is at the level that we have annotated PFAM, I'll see that this is the PFAM kinase domain corresponding to this ID, protein kinase domain. And the reason why it was labeled as varying is because, as you can see, this isoform here has a shorter CDS that kills one part of the domain. So this wouldn't be labeled as varying if I had selected the presence absence criteria, because every isoform in the gene has this type of PFAN domain, whereas when I select position, this tells me that there's variation, and I can go in and explore this. So I believe this is all I can show you for the, oh no, there's one other cool thing. If I run one position and presence analysis for a given ID, for instance, I did that for PFAM, I did the same thing for presence absence, I can view the combined results for that category. And this is a table that compares for every ID, the variation, the number of genes that are varying regarding each criteria. And if I click on the graphics button, I can view a result summary of that. And this will rank me all of the IDs and will build a plot where I can see the number of genes that are varying given its criteria. This is kind of useful to see whether total absence or partial disruption are more common, and in which category in which ID that happens. So this is kind of another cool feature that you have in the FDA analysis. And I think that with that, I'm done with the FDA. I don't know if maybe I should leave you a couple of minutes to play around, or if I should continue demonstrating, because there's not that much time left, like 7:30. So
yeah, we'll have another half an hour, right? Let's give them like 3 minutes.
cool.
Yeah, maybe like 20 minutes for you. And then a few minutes to wrap up. Yeah.
Okay. So yeah, what I think I'm gonna do is I'm gonna continue demonstrating. I hope you're being able to follow on your own computer while I do this. But if you're getting lost, just maybe watch me, and I'm happy to follow up on questions later via Slack. I'm gonna close all these. This is too much stuff. And we're gonna move on to the differential module. This has two main analyses, differential expression and differential isoform usage here. Differential expression is quite obvious. I'm not gonna go into details. It's a classic differential expression. You have two methods to choose from. And if you wanna know more about these, you can click on help and you'll have more info about the parameters and the differences between EdgeR and noise-seq, but I'm not gonna go into that. Since we're interested in isoforms, I'm going to run differential isoform expression. It's quite simple. There's only one method available. You need to select your significance level and whether or not you want to filter for minor isoforms. Minor isoforms are the isoforms within a gene that retain a low proportion of the expression of that gene. So it's very important to filter them out, because a very small change in expression of a minor isoform between conditions will very likely give you a significant DIU result, but it's actually a very residual change in expression and in expression distribution across isoforms that is happening. So we're not really interested in that. I'm gonna leave that as a default threshold. This will remove any isoforms that have less than 20% of the total expression of the gene, but you can make that higher or lower depending on your interests. If I select run, this will be very fast because we don't really have those many isoforms. If you ever encounter an error while running an analysis, this is the log, and this is where you'll be able to find the reason why, or maybe troubleshoot what's happening. Shouldn't take much longer. Keep in mind that you can run differential isoform usage. I did that for transcripts, but you can also do that for CDS. So we collapse the expression for transcripts that encode a synonymous isoform, that is that have the same coding sequence. And we repeat this type of analysis only considering the coding potential of the different isoforms. And it should be done soon. I'm going to show you the table for the IU proteins in the meantime. Here we have different types of, well, we have different columns. If you want to know more info about them, there's a help. Ooh, I think that ran out. Maybe the configuration is wrong, but you should be seeing a help button here, a help panel there. explain the parameters. Yes. So it's the same thing as for proteins. Basically, it gives you information about the variables contained in the table columns. And you can rank this by whether or not the gene was DIU, also do this by significance. And another piece of information that this analysis provides, it's not only whether or not there was a significant change in isoform expression for a given gene, but also whether or not there is major isoform switching, meaning that the most expressed isoform is changing between conditions. I'm going to show you an example of that. For instance, let's go to the gene I show you in the presentation. I right click, I select show gene data, and I click on the plus button here where I have all of the information layers available. I'm going to show you the expression charts. We have these at the gene, transcript, and CDS levels. And here's very clear why this was labeled as major isoform switching, because there's indeed a great change in expression. And it's so great that the most expressed isoform in each condition is different. And this is a good criteria to prioritize isoforms that might be undergoing, genes that might be undergoing strong splicing regulation. Another piece of information that we provide in the DIU analysis is the total usage change. This is kind of a mathematical magnitude that measures the amount of change that there was between conditions. That is the amount of expression that's being redistributed. It sort of serves as a proxy for fold change, but in this case considers every single isoform in the gene. And we have, if you click on this graphics button here and select view result summary, a summary will appear down here where we represent this total usage change in comparison to the log fold change of the gene. And we also show whether or not the gene was DIU. So this is kind of a nice summary. And normally those genes that are DIU and have isoform switching will have also a higher percentage in usage change. And the top genes in this plot will generally be interesting candidates for further study or perhaps for experimental validation if you're interested in that. So in comparison to fold change, for instance, we see here a couple of genes that don't change their gene expression much, but they do change isoform expression. This is a kind of an interesting observation because normally this means that this gene is mostly post-transcriptionally regulated, which is an extra point in favor of this candidate gene in comparison to genes that have a strong gene expression change. And I think this is it for the DIU. You will have the same kind of analysis for proteins, but in this case we do, as I said before, we collapse expression for transcripts that encode the same CDS. One interesting thing I wanted to show you too is how do we filter and export these tables, because perhaps I want to take this and move this into R and do my own type of plot or do some other comparison or intersect it with another thing. So if you click on this button here, it has sort of like a tick, you'll be able to remove or add row selections. For instance, I'm going to filter by DIU results, and I'm going to just select those genes that were detected as significantly DIU. I can change, I have this pop-up window that tells me how many rows I selected. I have 216 DIU genes. This is also shown here in the summary, DIU with or without major isoform switching and so on. And there is another cool thing is that you can change the significance level for the analysis. If you want to be more stringent, you make this lower and you will have less DIU genes. This is for filtering, and right down here, there's a button that will help you export. You can export the table, you can export only the gene IDs, and you can also export a ranked list with just the IDs and the significant values. And this is interesting because perhaps you want to run a gene set enrichment or something and you need this as input, and TAPAS interacts really well with any other tool that you may have external to it. So I think that's it for the DIU. I'm going to start running a DFI. Well, actually I have one run already. So the thing with DFI is, this is the third module, it's the feature analysis module, which is the one that integrates expression with functional notation to tell you whether there is a significant inclusion or exclusion change happening for a given functional element based on expression, right? You can run this for one or more categories. You can name this. For instance, say I wanted to just run this for PFAN domains. I click on clear all, I select PFAN, and I run it, right? You also want to filter minor isoforms, and because it has the same problem as before. And I can say DFI PFAN as the name. I can do this as many times as I want, and Tapas will store everything separately. Here's your help as well. And if I click on OK, it will start running. DFI takes kind of a long time. That's why I only selected run for one category. But I have already, luckily for us, run the DFI for all of the available categories. And here I have the results table. It's kind of similar to the DIU results. It will tell you whether or not there's differential feature inclusion for every single feature in a gene. If I rank by gene, you'll see that I have this gene here, for instance, these are the features that that are annotated, the feature categories. It will give you the category, the ID, and for every ID, it will tell you whether or not this is DFI, the significance estimate, and the favored condition, and whether or not there's major isoform switching. I'm going to move on to one that's more interesting, for instance, this one. And if I want to see the gene, I can do this, show gene data visualization. This will show me the graphical map. For instance, the gene has an NLS that has differential feature inclusion. If I want to check that, I can open the protein visualization, look for NLS, and see that it has actually a few NLSs, and there is an isoform that skips this whole part and has no NLS. So there is a change in inclusion, obviously. So if I want to see a summary of that, because say I have, in this case, I only have 2 isoforms, but when I have genes with a lot of isoforms, if I go to the expression chart, it might not be as obvious that there is a change. So Tapas summarizes that information via the gene feature ID visualization, where it collapses expression for feature including and feature excluding isoforms so that you can actually view the change. And it will also give you a magnitude of the change, which is similar to total usage change that I described before. So, well, lots of information. How do I summarize that, right? Because this is too complex, a table, too many rows. The DFI analysis has a summary table that I can open, and it will collapse the results by feature category. For instance, let's say I'm interested in nuclear localization signals. So I want to know how many DFI nuclear localization signals there are. Tapas will rank that for me, and now here it is. There are 43 DFI NLSs with different numbers of them being favored in neurons and in oligodendrocytes. And this is the number of feature IDs that were tested. If I want to drill down, I can see the genes that have that NLS and see the condition that is favored. I can also export this table, same as I did before. Any table that you see in Tapas can be exported. This is really convenient for any downstream analysis that you want to do. And there's also a way to see this as a summary. If I go to the main, I'm going to close this. If you go to the same main DFI table and you click on the graphics button, you will see a results summary appear. It takes a little bit, but it will eventually appear. And it provides some summary plots that tell you, in this case, the percentage of features for each category that are DFI in comparison to the proportion of features that are FDA that have. This compares the transcriptome's extent of variation, that is qualitatively how many times does this feature category vary within a gene, and then in comparison how many times does the gene have a significant change when I take into account the expression. this is a little bit complex to explain. You can go to the help and it will have a more detailed description. But basically what you want to look at is whether the bar is higher than the line. This means that this is significant or could be significantly DFI across the entire transcriptome in comparison to what you would expect in the background of the annotation. For that, we have a table. And if you look to PTMs, you'll see that actually there's a significant differential feature inclusion for that category across the entire transcriptome. So you might want to try and find a relationship between your experimental setting and this type of functional annotation. In this case, we have a neural mouse system, neural precursors versus oligodendrocytes. So this might be an interesting thing to consider when we want to interpret an splicing regulation in that experimental system. And I only have 10 minutes left. I don't know if I should just stop here or do one more thing and maybe then try and use the remaining minutes to wrap up. Liz and Anna, I don't know what you wanna do.
I'm fine with either.
Okay, so I'll just show you real quick one enrichment result and we'll wrap it up. As I said in my presentation, the differential module, but also any kind of result that you have generated in TAPAS can be input into a functional enrichment or a gene set enrichment analysis. And it's very easy to couple the differential module because these are ready here. If I wanna look at differential isoform usage genes, I can do that. I can select the background and the category that I wanna run the analysis for. I can click them all, which is what I would recommend doing if you wanna look at enrichment for any functional category in your DIU gene set. But you can also do that for differentially expressed genes, change the background, or you can input your own list. And this is where it gets interesting because let's say, as I said before, I'm interested in nuclear localization signals, and there is a number of genes that are DFI for that one category. I could output the list, input it here, and just run as an enrichment analysis to try and interpret it a bit more. I have already run a few. I'm going to show you the functional enrichment for DIU genes, which I labeled FEA DIU. And here I have for each feature category, I have a significance result, and I have a number of significant categories. As you can see, there are gene ontology terms. There are other categories, such as, for instance, these nuclearization signals, PTMs, polydenylation signals, some bar clip and RBP factors. There's a lot, right? So I can do a couple of interesting things. I'm going to show you that and just shut up and let Liz and Anna wrap up. One thing you can do is drill down, as I showed you before, and see which genes contain the NLS. That is, what is your target list gene that yielded significant enrichment in NLS. And you can also generate a summary here. You see, you click on view enriched features chart, you'll see this cool plot that shows you every single feature that is significantly enriched, the category it belongs to, the adjusted p-value that the analysis reported, and if you leave your mouse here, you'll see the number of tests and total items that you have in your transcriptome and the p-value, and you can export this image, and you can also adjust the display. Perhaps I want to show less feature IDs per category, so I click on one and I only see the most significant, and I can export this for my paper or do whatever. So as you can see, it's a very flexible, very complex tool. Rely on the help buttons, and if you have any questions while you're using it with your own data, please feel free to contact me or my colleague Pedro, who also works on TAPAS with me. And with that, I think I'm done. I'm gonna stop sharing and let Anna and this just wrap it up. If there are no questions, of course.
Let me check the platform. looks like we don't have any more questions. And all of the decks, Anna, Fran, and Angeles' deck. So I have put them. So by the way, Anna, Fran, and Angeles, I took your ISMB slide deck because I assumed they're exactly the same. And all I did was change the name to ECCB, and then they are now in the same Dropbox. So everybody should have access to all of them. You have any questions, please use the live Q&A or discussion. I think you can actually unmute yourself because I see some people are not muted. Yeah, if you have any questions, let us know.
Mm-hmm.
Has this session been useful for you? Maybe we should do that as a poll.
Yeah, we can do that.
I think this, yeah. Okay, let's do that.
Yeah, let's do that.
I'm pretty sure that there will be. Are you writing the poll, Liz? You're very quick.
It's on the platform because it's hard to have two screens on at once. Well, at least we got some feedback. Okay. All right.
I think this will shut down when the countdown is over. So we have like 6 minutes and then we're...
And then we will disappear from each other's lives.
We'll disappear. All right. I'll just, you know, since it's gonna shut down in 5 minutes, I'll just leave this on. If people want to talk.
You can switch on your video now if you want to say you know, to say hello or to give any feedback on this. Yeah. I mean, we are continuing working on this at present. Actually, we are working also on adapting Scanty for non-model organisms in a better way, and for those organisms that do not have a reference genome. So the classification and description is even more challenging. But you are welcome, you know, to provide also any feedback or input on features that you would like to see implemented in Scanty or in Tapas, or that you have, you know, any suggestions for improvement. You know, we like to know how people want to see their analysis done and what are their interests and their needs, so that we can provide tools for the community that are useful to everyone. So don't be afraid to ask for new things or you know, make suggestions.
Oh, you had a poll on tapas. We yeah, had people succeeded in getting tapas to run. Yes,
it's cool that at least 50% could. The 17% that couldn't, you can try and contact us via Slack or something, and we'll try and fix that.
How is Spain? I guess everybody is in Spain.
I don't think. I mean, not everybody here is in Spain. It's just a few of us.
That is nice.
I guess I'm just asking, is the weather hot? It's actually very cold right now in San Francisco.
Yeah. Well, here it's cooling down little by little.
Finally.
So 3 minutes.
Yeah. Did anybody?
So okay, I'm gonna make a poll. I'm gonna make a question before everybody leaves.
Yeah, because I understand that perhaps it's a bit intimidating to just unmute yourself here with 30 people and just say something.
Okay, I'm gonna say other questions. How many are you? Do you have your own ISO-seq data? Yes. No. Not yet, but soon.
I realize in Europe it's in the evening, so it feels very different probably for you because the day is, this is you know, in the evening, it's time to rest.
For me, my day is just starting. It's 11 a.m. now.
I wonder if there's anybody who joined from Australia or China or somewhere.
Well, that I think would be very, very painful. It's very late there.
Yeah, I was thinking that when I saw the schedule, I was like, it's not going to be possible for a lot of people to join. But well, I mean, you can't have it all, right? It's kind of hard to accommodate everybody.
So I created a question on the pool if you have your own ISO-Seq data. So I think that's an easy question for you guys. Right? So we can see that people are joining that are, you know, are just hoping to get their own data soon. So, but I don't know how it's possible. We have 4 votes and I have.
How can it be 25% times?
I don't know how it's 4 votes can have be split in 25 each.
You probably should have put that poll question up first instead of later.
Yeah.
Okay.
But we have 1 minute before we get kicked off.
Yes.
I think people are starting to log out.
Yes.
Yes.
We have 1 minute left.
Yeah. No, I think it's interesting to see that in general, more people, they don't have their own data. So this is more like, you know, an introduction of what to expect of what the possibilities are for the long reads. And I hope that with the presentation, you got a, you know, broad overview of how you can use, you know, long reads for transcriptomics analysis. And hopefully you will be convinced that this is a great way to analyze your data. Okay, so well, thank you everyone for joining and for being here. Thank you, Liz, and enjoy the rest of your day. And I guess we will talk again soon. And this is Angeles and Fran on the same screen now.
Here is easier to talk.
Okay. Bye-bye, guys. Thank you.
Bye.